So, hello, good evening, everyone, and welcome to the CENIEH. Thank you so much for joining us today in one of the most exciting, parties or festivities we have here at CENIEH. This is our birthday. This is our 13th birthday of the CENIEH in the complex of human evolution here in Burgos. We are happy and flourishing teenagers somehow with these 13 years old, but very beautiful and successful years where we have been together sharing many adventures and discoveries. And it is our 4th edition of our CENIEH Distinguished Annual Lecture. This is an event that we decided would be worth it to do to celebrate science and to make use of the opportunity to bring among us people who were the best of the best in their respective fields. We have been very successful so far of bringing today, in this special festivity, people who represent the most renowned and well-known experts in the world in the different disciplines that compose human evolutionary studies. And we are particularly happy today to have here with us Professor Robert Martin, Bob, as he is really well known everywhere. We almost think we don't even have to give a surname. He's Bob, everybody knows him. I mean, I feel even shy to have to introduce him because he does not really need introduction, but obviously he is, I was telling this morning, he is like what we call a pope in the field, in this case of primatology. And well, he has been the founder of many seminal contributions to the field of paleontology through the study of primatology. And also his studies have allowed us to really make very important inferences about human evolution and about issues that are crucial to understand the evolution of our lineage. In particular, today, his talk will be about one of the key events for the success of a species, which is human reproduction. He is well known for his activity, for his research, and he has a very, I would recommend to everyone, a very interesting and exciting book indeed about the topic he is going to be presenting today, which is the book, How We Do It, The Evolution and Future of Human Reproduction. And although I know you are all familiar with him, I just briefly introduce his profile. He has been, well, mostly developing his research and professional activity as director of the Anthropological Institute in Zürich. Then he moved to the Field Museum in Chicago in 2001. Then he was serving as provost for 5 years and then was curator of biological anthropology. And then in 2016, he was appointed an academic guest at the Institute of Evolutionary Medicine in Zurich. This is another, I would say, beautiful and interesting discipline he's bringing to us, which is evolutionary medicine, is this way of putting together the perspective of all the medical background that is necessary to fully understand the evolution of our lineage. And I think he's going to bring us very interesting insights. And well, I just want to thank you. It is a pleasure that you are here with us. You're really making this. We are putting the level so high that I don't know what we're going to do next year. Who can we invite to keep it up to the level? And it is a pleasure to have you here with us. So I don't want to make it longer. I just want you to give you the room. As you see, it's a full room. We are all very excited to have you. Well, welcome and feel at home because this is your home, Bob.
Thank you. I'm delighted to see so many people here this evening. Good evening to you all and welcome to this birthday party. This is the 4th lecture in this series and I feel very privileged to be here. Before I start the lecture, I just want to say a few words about some people here and also the buildings here at the center and particularly the museum next door. So first of all, I'd like to thank Mario Martinon Torres for inviting me to come here. It's the first time I've been in Burgos. It's a beautiful city and it's given me the opportunity to see what's going on here and it's quite stunning. And I'd also like to thank Kitina Moreno Torres, who has been very helpful on the organizational side and has made my visit here very easy. And yesterday I had the privilege of visiting both this center and the facilities, which are state-of-the-art. I haven't seen anything comparable to this anywhere. And so it's a major undertaking here. It's wonderful to see this level of commitment and funding for research into human evolution. And on the paleontological side, this all began with Emiliano Aguirre, who kicked things off with respect to finding key fossils. And after him came a triumvirate, a group of 3 co-directors, including Juan Luis Arzuaga, the director of the museum next door, Jose Maria Bermudez de Castro, and Oidal Carbonell. I probably mispronounced that, but these people together have started what has become a major enterprise. And I visited Atapuerca yesterday, and soon after the first discoveries of hominids here, I became very interested. It was clear to me 30 years ago or more that this was going to be a very important area for discovery. And this is undoubtedly the most important site in Europe for documentation of human evolution. And I visited yesterday. There are 10 different excavations going on, a total of 150 people. And more discoveries are coming out all the time. And so taken together, this institute and the site, the overall site of Atapuerca, is like a lighthouse in Zürich for our discipline for the study of human evolution. So I'd like to congratulate everybody concerned, and as far as this building is concerned, happy birthday. So with that said, I'd like to go straight on to my presentation. And as Maria said in her kind introduction, evolutionary medicine, or Darwinian medicine, is a particular discipline. It's relatively young. The first textbook only appeared in '95, but it has grown very rapidly. The basic idea is that in order to understand the human body and things that go wrong with it properly, you need to understand the evolutionary background. And in medicine, every person who does medicine should be taught the evolutionary background as well as the mechanics of keeping our bodies in shape. Maria is an excellent example. She started off in medicine, but all the time had the conviction that she would like to work in human evolution. And so she's managed to combine the two. And that is true of many people working in the field of biological anthropology. Now, I just want to quickly mention some basics about this. First of all, you can see there a picture which shows some of the 500 primate species that are now recognized. So it's a very large group of mammals. And my strategy initially was to study as many of them as I could, both in the field and in museums, to try to reconstruct the evolutionary history of primates. And my strategy was that we really needed to understand the evolution of the group to which we belong, the primates, in order to understand human evolution. The problem is, if you just look at humans and our closest relatives, the great apes, that's a very narrow perspective, and it's very difficult to test any ideas you may have. If you look across all primates, and all mammals as well, in fact, you can identify general principles that you can apply to the special case of human evolution. We are special in many ways, but we are very much a part of the evolutionary tree. Now, at this point, I just want to mention my book, which came out in 1990, which presents a lot of the basic evidence from all different fields. regarding the evolutionary tree of primates. And I'd like to, at this point, to pay tribute to my wife, Annalise, who's sitting there in the front row. She has been my constant companion and supporter throughout my career, essentially. And 3 days ago, we celebrated our 52nd wedding anniversary. And so I'd like to publicly thank her for all she's done to help me. And she also produced the best illustrations in that book are actually from Annalise. And Maria has already mentioned my popular book. That's not my choice of title, How We Do It. The publisher thought that was amusing. I didn't. But it's basically trying to do exactly what I set out to do. Take what I knew about the evolution of primates and apply that to the special case of humans. Maria mentioned that reproduction is actually a central topic when you look at it. Natural selection is differential reproduction. Some individuals in a population carry genes that help them to reproduce better than others, and that is what drives evolution. So we really ought to look at reproductive biology. The trouble is, there's very little that you can learn from the fossil record about this. So despite the fact that I'm standing here close to to the best paleontological site in Europe for human evolution, I won't be able to say anything about fossils. I'm going to be talking about inferences we can make from studying modern humans. It's a vast topic, and so I've chosen 3 subtopics to talk about as illustrations of applying evolutionary principles in the hope of getting guidelines for human medicine. The first is sperm biology. The second is the evolution of human birth. And then the last one is the evolution of life histories, including overall lifespan. So starting with the first one, sperm biology, one of the first questions that we need to ask, not just for humans, but all mammals, in fact, is why does an ejaculate need to contain so many sperms? On average, for the human male, the ejaculate contains 250 million sperm. That is actually a reduction, as you will learn, to what it was 50 years ago. But 250 million is still quite a substantial number, and you only need 1 sperm to fertilize an egg. In fact, if more than 1 sperm fertilizes the egg, it's disastrous. And so why so many sperms? That is the question. And one idea that's been at the forefront for a long time is competition. It's the idea that, first of all, that you have competition between sperms. And the bottom right, you can see an illustration. The fertilization has been seen as a marathon with 250 million participants released, and only one of them could be the winner. And that winner is somehow the best out of those 250 million sperm. I don't see how any way in which that can be true. The DNA, the genetic material in the sperm head, is essentially crystalline. It's not active until fertilization occurs. So it's very difficult to see how sperm could be selected by competing with one another because the characters are not externally visible. Now, I want to give you an example now of how comparative biology can give us a different perspective on things. Because one entrenched idea has been that you have this massive race, that the sperm are swimming all as fast as they possibly can, and the best swimmer wins the prize of fertilization. Now, it's very simple to demonstrate this cannot possibly be true. If you look at the top left, you can see there a size comparison between a mouse, a human, and a blue whale, the largest mammal alive today. At the bottom, to scale are 3 eggs. I mean to scale compared to one another. The egg of the mouse is actually slightly larger than the other two, slightly smaller than the other two, but humans have an egg that's slightly bigger than in the blue whale. Because in evolution, increase in size does not involve bigger cells, it involves more cells. Okay, that is what makes the difference between a blue whale and us. They have vastly more cells than we do, but each individual cell is pretty much the same. In the bottom graph, you can see the size of egg cells for various mammals. Those are the blue points, and the 3 red points are primates, humans, one of them, and 2 other non-human primates. And you can see that as plotted against body size, there is no overall increase. Essentially, the size is remaining more or less constant right across that size range. Well, in fact, the same is true right up to the blue whale. And a blue whale actually has a slightly smaller egg than we do. At the top right, you can see the heart of a blue whale compared to a human being. The heart of a blue whale is roughly as big as a human being in height. And in fact, an individual testicle of a blue whale is as big as a human being. And the ejaculate contains 20 liters of semen. And so this is a vastly different problem. Now, it's possible that in a mouse, the sperm could swim all the way up the tract. But if you take a blue whale, the tract is 100 times longer, you have sperm of exactly the same size swimming up the tract. And there's no way that they can use the same swimming mechanism to swim so much further. And in fact, there is a medical intervention called Cintography, which is used to diagnose medical problems. And the basis of the technique is that you insert labeled radioactive labeled particles into the vagina, and they are transported passively through the vagina, through the womb, and up into the oviducts. And so here on the left-hand side, you can see an image, and the dark areas are where radioactivity is detected. So these are inert particles, they cannot swim, they're being carried up the tract in humans, and this is actually used for medical diagnosis. If you look at the image on the right, in fact, that particular woman had a blocked left oviduct, so you can see no label showing up there because the sperm simply couldn't swim up that far. So they all swim to the right side. Now, so the idea of a swimming competition can be forgotten. And so we're left with the question, why do you need so many sperm? And associated with that is the question, are they all needed? Do you actually need to have that sort of number? And the answer is yes. So if we look at this graph, if you look on the left and the right, if you look at the top in each one, what you have up this axis is the likelihood of pregnancy occurring in the course of a year according to sperm concentration. And so you can see here that it gradually climbs and then levels off. So about this level here of 50 million sperm, you get an optimal level with about 25% success rate. But anything below that, 50 million per mil, is insufficient to get that level of success. And if you look here, the axis here is rather different. This is not the concentration, but the total count. The average human ejaculate contains 3 milliliters, not 20 liters like a blue whale, 3 milliliters. then what you need to do is to multiply these figures by 3, and that gives you the total sperm count. So what you can see here is that about 150 to 200 is the point where you reach the plateau. So anything above that, you don't need more sperm to get pregnant. But that is 150 million sperm out of the 250 million that are required for frequent pregnancy. Now, so far I've been talking about needing large numbers of sperms, but there's another point to this, and it took me a while to grasp this, and I didn't actually know about this while I was writing my book. It's something I discovered subsequently, which is that you can have too many sperms. And this was discovered, for example, with in vitro fertilization. When you fertilize an egg in a test tube, you do not want too many sperms around, because if that is the case, you can get fertilization by more than one sperm. And commonly you can find fertilization by 2, sometimes 3, if you have too many sperm around the egg. So you need a certain number to ensure pregnancy, but if you have too many, it can be disastrous, because if you have a fetus that has too many chromosomes because of the additional sperm or 2, that is always fatal. And so natural selection should eliminate that possibility. And I think this is why you get this picture here, which is that the female tract, as far as sperm are concerned, is like an obstacle course. The sperm are deposited here in the vagina, and here is the neck of the womb, or the cervix, and that serves as a filter. There are strands of mucus, and abnormal sperm do not make it through the cervix. So that is the first filter to remove sperm that are not properly formed. Then the sperm migrate through the womb and into one of the oviducts, and the passage into the oviduct is another filter. And then after passage through the oviduct, the sperm attach, if they're healthy, they attach to the lining of the oviduct for a while, and then they're released. And at that point, a sperm can fertilize the egg. But of the 250 million that start here, at any time, only about 200 sperm will actually surround the egg. So this whole system is set up to filter out damaged sperm and to ensure that only a small number surround the egg at any one time. And so this is why you have the vast number. The male is producing vast number of sperm in order to ensure that enough will get close to the egg. So this is a very different picture we're seeing now, and unfortunately in medicine, ideas have been dominated by the view that sperm migrating up the tract is some kind of marathon race, and it's not. It's a passive thing mostly. The sperm are passively carried up most of the way. Their swimming is largely when they get close to the egg. That's when the swimming really comes into play. But it's a very different system. Now, in recent years, it has become absolutely clear that there is a major medical problem in that the number of sperm in the average human ejaculate has been falling steadily for something like 50 years or more. And you can see in this graph here, it starts in 1930 here at the bottom, and it goes across to 1990. That's a 60-year period. And sperm counts from various studies over those years have declined by about a factor of a half. So here you have a starting point, and down here you've got about half the total sperm count that was true 50 years or so ago. But this was quite controversial because a lot of people pointed out that there aren't many data points here, and many people suggested this is actually a plateau here with a drop and then another plateau. And it was suggested that this is because the method for changing sperm counts, for measuring sperm counts, changed by the World Health Organization at this time. And that's true. But we have a brilliant example of comparative study here is because a vet in Australia said, well, if it's true, we use those same methods for counting sperm in domestic mammals. And the author showed that in bulls and in pigs and in sheep, sperm counts have not changed over this same period. So whatever is going on here, it affects human beings, and it's not affecting farm animals. And more data were collected, and so this paper was published in 2000. By 2017, far more data were available, and you can see there are a lot more studies have been picked up from earlier on, and it's statistically highly significant. And this is an extremely important and very careful paper, and I have absolutely no doubt that sperm counts are actually declining. And again, this is roughly a halving in numbers. Now, that's still, the average is still around 250 million sperm, so it's enough for fertilization on average. But what this decline means is that more and more men are going to fall in this range where fertilization becomes more difficult. And we are seeing increasing problems with fertility in the industrialized countries. Now, Shanna Swan, who is an author on both of these studies I've shown you, recently wrote a book called Countdown, a very neat title, it says it all. And in this book, which came out just last year, she reviewed lots of evidence, and I think convincingly so. And she, like other people in the field, has come to the conclusion that a major factor here is what are called endocrine disruptors. Endocrine glands are glands that produce hormones in the body, and so endocrine disruptors are organic chemicals in the environment that mimic the effects of hormones, and that is where the problem arises. And there's quite a lot of evidence that it's endocrine disruptors that are contributing to this decline in sperm counts. Now, if we look at the endocrine glands, these hormone-producing glands across vertebrates, and so this is a comparison between human on the left and fish on the right. All of those organs listed down the middle produce steroid hormones, and they're present in fish. So the same system has been present for 450 million years at least. And so it's a very ancient system. And our hormone system is very similar still. Now, the important thing here is that these are different steroid hormones. And at the top and bottom are reproductive hormones. You have testosterone and estrogen, such as estradiol. And here you have progesterone, which is important in pregnancy. And the basic structure that gives rise to these is a substance called pregnenolone. And you can see that every single steroid hormone has 4 carbon rings. And that was thought at one time to be essential, that in order for a substance to behave like a steroid hormone, it had to have those 4 carbon rings. And the difference between these hormones is rather small in chemical terms. It depends on the side groups, which differ from hormone to hormone, but it's a relatively minor difference. But the unusual thing here comes out of research that was conducted by Charles Dodd in England, and he devoted his life to the search for an artificial estrogen. At the time when he started, about 1930, it was very expensive to synthesize steroid hormones, and so he took various organic chemicals from the industry, the petroleum industry, and tested them. His basic approach was to remove the ovaries from a female mouse, and so her reproductive system became inactive, and then he would feed the mouse with one of these chemicals, and if reproductive activity started up in the tract, that would indicate that the chemical substance was acting like an estrogen. And so one of the first substances he looked at is Bisphenol A, generally known as BPA. And this chemical is widely distributed in our environment now. It's used to harden plastic bottles. It's used to line tin cans holding food. It's used in epoxy resin. For instance, if you have a dental filling now, it's glued into place with epoxy resin. And if somebody measured your blood here from all of you, most of you, perhaps all of you, will have at least some BPA circulating in your bodies. And the reason Charles Dodd was interested in this is back in 1930, he discovered that BPA acted like an estrogen. So if you took a female mouse without ovaries, made her eat some of this, her female tract would start operating again. For whatever reason, the industry decided to use BPA for all of those uses today. But he actually lost interest in BPA because he found a substance, diethyl stilbestrol, which you may know as DES. It was administered to millions of pregnant women as an artificial estrogen, a substitute for natural estrogen. And eventually it was shown that it was causing huge damage. And this is transmitted down generations. In the United States, there is an organization called DES Daughters who were in the womb at the time their mothers took this DES, and their organs were damaged, and it goes on through the generations. There are now DES granddaughters and even DES great-granddaughters. That effect is still there 3 generations down the line. Now, both BPA and DES just have 2 carbon rings, whereas a typical steroid hormone, such as the estrogen estradiol, has 4, as I pointed out. So chemically, these 2 substances don't look like anything like an estrogen. BPA is active in an estrogen-like way. DES was very active, and that's why it was such a problem eventually. Now, here you need to have an evolutionary background. The reason this is happening is because the organic chemicals that Dodds was trying out do not occur in nature. And it's the side groups that happen to trigger the receptors on the cell wall for the steroid hormones. And no organism became adapted to that because these chemicals did not occur in nature. And so by using and releasing them in large quantities, we are flooding the natural environment with chemicals that are mimicking the effects of steroid hormones. And this is a major problem. And just quickly, I want to show you, this is a study in China. In China, the controls on production in factories are quite limited. And so you can find factories that have huge amounts of BPA in the workplace, and that ranges down to factories with no BPA present. And what they did was to simply examine the semen of men working in those different factories. And you find a statistically significant relationship between the level of BPA and the decline in sperm semen quality. The figures outlined in red there show statistical significance for concentration of sperm and total sperm count, but not for the volume of the ejaculate or sperm morphology at the bottom. But you can see there pretty massive effects of BPA in the workplace on semen counts. And if we look at the female side, this is actually an example from in vitro fertilization, IVF. And in order to perform that, a woman receives hormone treatment to trigger ovulation, and egg cells are collected. And what the diagram, the histogram at the bottom shows, these are what are called quartiles. This is the first fourth of the data, second, third, and fourth, so it's split into 4 groups. And they are 4 groups with increasing BPA. And you can see as BPA increases, the quality of egg retrieval declines in all categories of how many oocytes and whether they're fertilized and so on. And you can see again, there are significant relationships between these effects and BPA levels. So it is absolutely clear to me that we have a major environmental problem. And so this is just one way in which the evolutionary perspective can alert us to medically important features. Okay, now I want to carry on now with the second category, which is the evolution of human birth. And a key point about human birth is that it is very challenging. Half of the audience here will have been through this problem probably, or about to do so. And giving birth is a great challenge. And the reason for this is because the pelvis is relatively small compared to the size of the baby's head, the newborn baby's head. And what you can see here on the left is the passage of the neonate, the newborn, through the female tract during birth. And so you start at A at the top and go down to D, and then E, right down to H on the left. And what happens is that the fit is so tight that going into the pelvic canal, the baby's head is rotated 90 degrees, so it's looking to one side. And then to go out at the other end, it rotates another 90 degrees. And so the baby comes out looking backwards and not forwards. Every other living primate gives birth in a simple throughput, and the baby's head is facing forwards when it comes out. So this is a unique feature of humans. There's a very tight fit between the newborn's head and the pelvis, the pelvic canal. Now, all of this is summed up in what Sherry Washburn called the obstetrical dilemma. And basically the idea here is that in women, there is a trade-off between 2 different requirements. For walking upright, you need the pelvis to be as narrow as possible from side to side. But to give birth easily, you want the pelvis to be as wide as possible. So what's happening in the evolution of the female pelvis is a compromise between those 2 opposing influences. In men, there is only one requirement, which is walking upright, and so men do not have to have that compromise. And so Sherry Washburn introduced this term obstetrical dilemma, which is still used today and is a key feature of human birth. And a lot of women still today die in the process of birth because the neonate gets blocked in the pelvic canal. And Washburn neatly summarized this idea, but he ended up by saying that the obstetrical dilemma was solved by delivery of the newborn at a much earlier stage of development. Now, this is true in one sense, but it's turned out to be very misleading, and I want to show you why that is, because there's something unique about the human birth process, but our babies are not born earlier than you might expect, except in one limited respect. So if we look at the pelvis now, on the left, the top, you can see a typical male pelvis, and the symphysis, the join between the two parts of the front, is deep, and there is a pretty narrow angle beneath that join. If you look at a female pelvis, the join is much shorter, and there's a wider angle. If you look in from above, this is the birth canal in a female pelvis, and it's round, and this part of the vertebral column has been pushed back as far as possible out of the way to leave a pretty circular canal. Whereas in men, there's no requirement that the sacrum, that part of the vertebral column, protrudes into the space here, and the profile is more heart-shaped than circular, as in a female pelvis. And so there is sexual dimorphism in the pelvis, and this is unique to humans, this extreme degree of sexual dimorphism. One thing I want to mention at this point is that Juan Luis Arzuaga, in fact, did his PhD thesis on sexual dimorphism in the pelvis, and did an extremely detailed analysis of the differences between men and women in the pelvic anatomy, and published a paper which I still find the most useful thing in the literature on this important topic. But what I want to do now is address this claim by Washburn that human infants were born at an early stage of development. Now, this looks a little complex, but it's easy to explain. Along this axis, you have the amount of fetal brain growth corrected for body size of the mother. And along this axis, you have the amount of brain growth after birth. Okay. And you can see for non-human primates generally, there's a negative relationship. The more fetal brain growth you have, the less postnatal growth. And that's a fairly obvious thing. But here's humans. We are outliers. And we are outliers even with respect to fetal brain growth. A lot of fetal brain growth occurs during human pregnancy. But the point is our adult brains are so big compared to other primates that you need an extreme amount of brain growth after birth. And so in fact, we're not born immature relative to other primates. We have a lot of postnatal brain growth. But what it means is that the brain at birth in a human baby is immature compared to other primates. And that makes our babies helpless compared to, say, a rhesus monkey or a chimpanzee. And if you look at brain growth in non-human primates, you see this picture here. The top right is for a macaque, and this is brain weight against body weight for a developing fetus here. And you can see there's a very fast growth of the brain here up to this point, and then birth occurs. And the brain growth slows down relative to the fetal pattern. And adult brain size is only about 2 times the size at birth. Whereas in humans, and this is completely unique, no other mammal shows this pattern. We show this fetal pattern of brain growth. Birth occurs here. Growth doesn't slow down. The brain continues to grow at the fast fetal rate for 12 months after birth. And Adolf Portman in Switzerland, who worked on this problem, said that human pregnancy is actually 21 months, 9 months in the womb, and 12 months outside. So human babies are relatively immature compared to adulthood, and it actually takes much longer for adult brain size obtained. And adult brain size in humans is about 4 times the weight at birth, not twice the weight as it is in all non-human primates. And so essentially what Washburn was arguing, and I totally agree, is that in humans, by the time brains have reached this size at birth, they will only just pass through the pelvis. In order to attain larger brain sizes, you have to have a continued pattern of fetal brain growth for the first year of life. It's quite interesting that our babies don't start to walk in a bipedal fashion until after about a year, and they don't start to talk for the first year. So in terms of the central nervous system, this is a period of immaturity. So Washburn was right in that sense. But in fact, human babies are not born earlier than in great apes in any general sense. But his basic argument was that the pelvic canal is a constraint on the size of the human baby at birth. And it is for that reason that you see that special pattern. So, if we could find a mammal that has a big brain similar to ours, a body size similar to ours, but no pelvis, I would expect that they would give birth to a baby twice as big as ours, with a brain half of the adult size. If it's true that it's the pelvis that's constraining the size of the brain at birth. And in fact, mammals like that do exist. Whales and dolphins particularly, they have no pelvis. They have 2 little tiny splints. That's all that's left of the pelvic girdle. So there's no constraint on the size of the baby at birth. And there are dolphins that are about our body size. Their pregnancy is about 10 months instead of 9, but they give birth to a 7-kilogram baby with a brain half of the adult brain size. So they can do it because there is no constraint on birth. We can't. So our babies have to continue our development of fetal brain features after birth. And so recently I tried to put this together in an overall summary, taking an evolutionary tree that shows the relationship between apes and humans. And here we have gibbons. And I think gibbons are dwarf forms. I think they reduce their body size in evolving from the common ancestor. Here and then here you have a common ancestor for great apes. And humans, and here, these are the African great apes, gorilla and chimpanzee, and then human. And what I have above here is, in red, is the adult brain size. And on the icon itself, you can see the adult body weight. And then in blue, you see the data for the newborn. And the pregnancy period in great apes is quite long. It's almost as long as in us, but it's 8.5 months in orangs and gorillas, and it's only 8 months in a chimpanzee, whereas it's 9 months in humans. And here you can see body weight now. The average body weight of a human baby at birth is about 3.5 kilograms, whereas the average for great apes is just over 2 kilograms. Our babies are almost twice as big as they are in great apes. So no way are they born early. In terms of overall size, they're actually born more developed. But one very interesting thing here that I only spotted when I produced the diagram for this lecture is that if you look here, that is the adult brain size of a chimpanzee, 385 grams. And that is the size of the brain of a human neonate. So a woman gives birth to a newborn whose brain is as big as in an adult chimpanzee. So this puts it in perspective. We're not giving birth early in any sense. The reason that pregnancy has to be extended for 12 months after birth, essentially, is because of that pelvic constraint. And this is a major concept. I work as, I have a guest position, as Maria pointed out, in the Institute of Evolutionary Medicine in Zürich, and there is a group there with which I'm associated, which is looking at the pelvic constraint in fossil hominids as well as in modern humans and great apes. So, I now come to the third and last part of the lecture, and I want to take that same comparative approach to life histories and overall lifespan. So I'll be looking at the human life cycle, and interestingly, it has been said in literature, Shakespeare said this of men, that there are 7 ages of people, and this is a painting by Hans Baldwin Green from 1544 that shows the 7 ages of women. That is actually the title of the painting. So you start with the child down here, and these stages are approximately 10 years apart, because at that point 70 years was about the average life expectancy. That has increased, as I will mention later. And what we can do is take this and look at it in strictly biological terms. And you can see there is actually biological evidence underpinning that division into 7 stages. First is infancy, then there are 2 parts of childhood, and then you have a juvenile period, adolescence, adulthood, which goes up to 70 years, and then after 70 you have senescence, where various problems of use of the body appear. And at the bottom here you can see the dental change. There's a milk set and a permanent set, and so significant milestones are replacement of the milk teeth by adult teeth, and particularly the 3 molar teeth at the back of each tooth row. And you can see here that the eruption of the 3 molar teeth actually coincides with the boundaries between these stages. And the milk dentition is complete at the age of 3 here, and weaning occurs between these 2 markers. So there are actually dental markers that indicate those phases of human development. Now we can look comparatively at various aspects of life histories across mammals. So all of these black points are mammals of various kinds. And primates are shown in red, and the green points are bats. Now this first graph shows the age of first reproduction. So this is essentially the age of sexual maturity plotted against body size. And you can see for their body size, bats and primates achieve sexual maturity very late. So we have a slow life history. And this is maximal lifespan. And what we see here is that once again, bats and primates are outliers. We have relatively long lifespans compared to most mammals. And these 2 things go together. Mammal species with long lifespans achieve sexual maturity later in life. And what this translates is the opposite effect when you look at overall fertility, how many babies can be produced and raised. And you see that declines with increasing body size. This is annual fertility, so how many babies on average can be produced per year, so it's the rate of reproduction. And in this case, bats here and primates lie below the line. And so our reproductive productivity is lower for any given body size. And so these things go together, and it's also well established that large brain sizes are typically associated with relatively long lifespans. And humans are an extreme case. We have extremely large brains, and we have an extremely long lifespan, averaging about, is a maximum for us, turns out at about 125 years. Nobody has yet lived beyond that. The question is that some biological barrier, or can medicine enable us to live longer than that if we want to? Now, this brings me to a unique feature among primates of the life cycle in women. At the age of about 50, women become infertile, and that is called menopause. And it's often been said to be unique to humans. And that was long the case. There were no examples from other mammals at all. But that has changed, as I will show you. There is a map of the world showing menopause, the age of menopause around the world. And the average for each continental area lies between 46 and 52 years. So it's a very tight range. On average, it's about 50 around the world. So this is something very precise. It's something that is a biological process, and it's not very variable at all. And so this indicates to me that there is some fundamental reason for that. But many people have tried to explain why we have menopause and no other primate species does. And one of them is the grandmother hypothesis. It's very popular. And the idea is that having reached a certain age, it's more genetically rewarding for a woman to stop giving birth herself and to look after her grandchildren. And so this is the idea. And there are some data that indicate that, for example, for 2 samples in Finland and Canada, you can see that there's a general upward trend. The number of surviving grandchildren increases as the lifespan of a woman increases. So long-lived women, on average, have more surviving grandchildren. And so this kind of indirect evidence has been used to support this idea. It sounds very appealing, except to me. I'm personally offended by the idea that there is no selection on grandfathers. And I did a word check on the basic paper launching this theory, and there's no mention of grandfathers anywhere in the paper. Okay? Now, most of the arguments that are being applied to grandmothers here apply equally well to grandfathers. And if it doesn't, then why don't men die at 50? Because there's no point in having them around. The age of men and women in a monogamous relationship tends to be roughly the same. And so an old woman is living with an old man. And if she stops breeding, what do we need men for anymore? So if we look at the basic mechanics of this, a widespread idea is that menopause is when a woman's ovaries, left and right, run out of eggs. And there's a so-called menopause threshold, which is that when you fall below a level of 1,000 egg cells in the ovaries, that is when menopause occurs. And so here you can see a graph. There's here an image of a young ovary and an old ovary where starter egg cells are few and far between. And here you can see the decline in the number of egg cells in an ovary, and that is that menopause threshold at about 1,000 eggs. Now, here again, comparative evidence is very helpful. Here we have comparative data from human ovaries and chimpanzee ovaries. And so the points indicate the number of starter egg cells available. And you can see in chimpanzees there is a simple linear decline. The older a female chimpanzee gets, the less eggs she has in her ovary. But she still has quite a few by the time she reaches 50. But 50 is about the upper limit for breeding in chimpanzees. But on average, they don't live much longer than that. Whereas you see the picture in the human female above with the red points, and you can see something you're not seeing in chimpanzees, which is the graph accelerates. Second half, in fact, a mathematical analysis indicates a slow decline here and then a fast decline after the age of 37. This suggests to me that natural selection is actively increasing the rate of loss of egg cells up to the age of 50, which is where menopause occurs. And this is not seen in the chimpanzee. Now, some people have claimed that menopause is not unique to humans. And in fact, there's one study suggested that you have menopause lasting a few years in rhesus macaques, and in chimpanzees you have a rather short period. Here the maximum recorded lifespan is 60 years, and I've said the ovary runs out of eggs at about 50, and or maybe a bit later. In humans, this says 122, I think it's about 125 for the overall lifespan. But even if this is menopause in non-human primates, it's much shorter than in humans. If a woman reaches the maximum lifespan of 125 years, she spends more than 2/3 of her life after puberty infertile. So that is really unique. But in fact, these data are based on simple one-off studies. And it's been pointed out since what we need is a population measure, is you look at all females in a population and you work out what the likelihood is that a female can continue breeding after a certain age. And so this is called, a method you can see at the top there. Oh, sorry, my fingers are getting a bit tired. At the top there, the female reproductive state. Green is reproductive. That's the period during which a female can become pregnant, give birth, and post-reproductive is menopause for the population. And this is associated with a statistical test. And so in all of these cases where you see a fairly big red part, that is statistical evidence for menopause. So it's relatively new that in the whale group, there are several species that have menopause. So it's not unique to humans. And if we look at two whales here, this is something very interesting. Here are two sister species. They replace each other geographically. This is the long-finned pilot whale, which shows ovarian activity right up to the end of life. And here you have the pregnancy rate in mauve, and red is ovarian activity. It's declining, but it's still relatively high. In the sister species, on the right here, you can see that there is a definite decline as the maximum age approaches. If you look at pregnancy rate, by around 35 to 40, that has dropped essentially to zero, and ovarian activity ceases by the age of 50. So you have menopause in short-finned pilot whales, but no menopause in long-finned pilot whales. Now, if we want to know why our species has menopause in females, we really need to study these two species and see what the difference is that is associated with that. It's not grandmothering. And the same is true of elephants. It's recently emerged that the Indian elephant, the Asian elephant, has menopause. So there are very few Indian elephants that give birth after the age of 50, as you can see in the bottom left diagram. The African elephant doesn't show that decline. So we have menopause in the Asian elephant, but not in the African elephant. So we really need to study those two species to see why we have menopause in one and not in the other. Now, all of this is related to a major difference between men and women, between males and females in the formation of sex cells. And in this diagram on the left, you have the female system here, and this is childhood, and here is puberty for both females and males. And it's very interesting. Midway through pregnancy, a female fetus has 7 million starter cells in the ovary. By the time she's born, that's been reduced to 2 million. Nobody really has explained why that is. But at puberty, a female is starting off with under 1 million starter cells. And then there is the first cell division to produce an egg. And that doesn't continue to the second division until fertilization occurs. And then you get the egg. The final division occurs here as fertilization occurs. Whereas in the male side, you get continuous cell division to produce sperm. As once puberty has been reached, you get cell division after cell division to maintain that huge production of sperm. Now, the important point here is that mutations primarily occur during cell division. So the more cell divisions you have, the more mutations you're going to find. And this really wasn't understood until recently, and with modern techniques it proved possible to actually count the number of mutations in sperm and in eggs. And this is the result. For sperm, the blue dots, you can see there's a much higher rate of mutation than in eggs in red. And you know, we've heard an awful lot about women having a certain age at which it's optimum for them to reproduce, and that problems arise with a woman's age. But as I pointed out, in couples, the male is going to be about the same sort of age. So if you have an old female, you have an old male. And in fact, the male is far more dangerous from a point of view of mutations than the female, as you can see from this graph. The difference is about 4-fold later in life. A male has 4 times as many mutations in any of his sperm than a human egg. But even in the egg, you can see there's a gradual increase in accumulated mutations. And it was only measured up to around 47 years of age. But eventually, there are going to be a lot of mutations in eggs as well. Now, I want to finish off with just one final question. Why do we die? And from a biological point of view, that's difficult to understand. You would expect natural selection to keep any individual going as long as possible and reproducing as long as possible. The longer that individual lives, the higher the chance of leaving genes in the next generation. So why do we die? And all kinds of possibilities have been suggested for that. And one of them is the increase in mutational load, that as we age, we accumulate mutations, and that increases the risk of things like cancer and so forth, which again are largely linked to accumulating mutations. And so the reason we the reason we die eventually is because we've accumulated so many mutations that we're victim to disease. And until recently, it was generally believed that there was no difference between mammal species, that the number of accumulated mutations depends on how long you've lived. So in a mouse living 3 years, the expectation was that humans would accumulate the same number of mutations in 3 years as a mouse. And that's turned out not to be true. And this is a really huge finding. It just came out earlier this year, which is that if you look at mean mutation rate, which is that axis, against lifespan, as lifespan increases, you find that the mutation accumulation rate decreases. So a human living for 85 years, as in that diagram, on average, accumulates only a fraction of the mutations that a mouse does within a short lifespan of maybe 2 or 3 years. So there must be mechanisms in long-lived species to reduce the accumulation of mutations. And there are known mechanisms to correct genetic damage. And it looks like one of these trade-offs, again, that if you're a short-lived species, you do not invest in correcting the genetic damages, because it doesn't matter. If you're a long-lived species, accumulation of mutations is going to become a big problem. So you invest in mechanisms which cost energy to reduce the age, the rate of accumulation. And so if this is referred to as mutation burden, and in each of these diagrams, this is birth and this is the end of life, and the color increase illustrates the increase in mutation. Here we have a mouse that by the lifespan of 3 years has already developed a high number of mutations, but humans do not reach that point until they're 85 years of age. And so there are mechanisms, probably genetic repair mechanisms, that in long-lived species like humans are reducing the accumulation of mutations. Now, this really gives some hope that we might be able to have medical treatments to reduce the effects of age. I personally think it's highly unlikely we will ever be able to extend the maximum lifespan beyond 125 years. Some biologists think that's possible. I don't. I think there are so many trade-offs that that would be a disastrous thing to aim for, and I'm not sure it would actually be very helpful for society. The important thing for individuals is how many of those lives you live, how many of those years you live in your life are healthy. This is called the health span in evolutionary medicine. It's the period of years for which you enjoy relatively good health. And so what I would hope for medicine is not that my lifespan will be extended to beyond 125 years, but my lifespan between now and whatever the end may be is as healthy as possible. So that's my take-home message. Thank you for coming.
Thank you, Bob, for a wonderful, wonderful and enlightening lecture. I think you have tackled like many interesting topics that I think are of interest for all of us. So thank you. It has been a wonderful choice to have you here today. So now it's time for anyone who wants to ask questions about this very interesting thing about humans. I think, yeah, there's one thing. So you pass.
Can you hear me? Should I ask in English directly?
You can ask, and I will translate, or if necessary, or in English.
Okay.
Yeah, that's okay. Yeah.
Thank you very much. First of all, that was very. It was a beautiful talk, very clear. My question is in regard to the obstetrical dilemma. You mentioned a trade-off between the pelvis size, where if it gets larger, it would be an issue for bipedal locomotion. And so I was wondering if there are any experiments on how large a pelvis could get before becoming an issue in bipedal locomotion. Is there something like that?
Yes, I'm not sure if I can answer that directly, but there is quite a bit of evidence. One, I think this is relevant to your question. There's been a study of the change in the pelvis over a woman's life. Okay, this is an average graph. What it shows is that over a woman's life, the size of the pelvic canal is maximized. And it peaks at the peak reproductive age of around 30. And then after the age of 50, there's a decline in pelvic canal size. It no longer matters because fertility has ceased. So that's one very interesting piece of evidence. But I'm not sure whether that answers the... have I missed something there?
No, yes, he's asking if there are studies that explain what is going to be the limit of how much large the pelvic inlet could be without compromising locomotion, if there are specific studies.
Right, yeah.
So
that's a very good point, because you mentioned the trade-off between locomotion and birth. And the one important point here, as I said to Maria earlier, I have fallen into the trap of, I look at modern humans and I see something like the obstetrical dilemma. And so I try to explain that in evolutionary terms. But I'm assuming that 30,000 years ago, when we were hunters and gatherers, that things were the same. So if I talk about an obstetrical dilemma, I'm assuming it was the same in hunters and gatherers. But the thing is that since we started living in settled communities 10,000 years ago, our skeletons have become smaller and more gracile. And I think that has probably reduced the average size of the birth canal. And so I think this has made things worse. So because I often ask, why in evolution should it happen that birth should become so challenging that the shoulders might get stuck or the head might get stuck? But if during the last 10,000 years we have changed our morphology, our anatomy, that might explain. So I think a lot of medical conditions can be explained by that factor that living in settlements wasn't actually a very good idea. Turning to agriculture was not a very good idea in the long term because it brought about changes. And the basic tenet of evolutionary medicine is this mismatch. Idea is that we evolved for a hunting and gathering existence. 10,000 years ago we began to settle down, but that's not enough time for us to adapt it. So I think that should answer your question.
If I may, I guess the actual question is, if it was shown that the pelvis of women could be enlarged further without compromising bipedal locomotion, maybe there could be an alternative hypothesis, like an energetic competition between the baby and the mother. So it is in the interest of the mother to reduce interbirth interval as much as possible to have more children. So I don't know if I'm explaining myself, but I'm guessing what I'm saying is the baby wants to stay as long as possible to be more, to have a higher chance of surviving, and the mother wants to have another baby as soon as possible.
That's absolutely true. There's quite a lot of discussion about paternal imprinting of genes. And the father's aim is to have the baby grow as big as possible, regardless of the woman's body. A woman's body is adapted, as you say, for future reproduction. She wants a big baby, but not so big that it endangers future reproduction. So this is, again, one of these trade-offs here between the sexes. So that's a large area of research, is that difference between male and female. I think you were also asking about the maximal size of the hips, because a relatively easy solution is to the obstetrical dilemma is to have a larger pelvis, and then the baby comes out fairly quickly. So why doesn't that happen? And it looks as though it's because of locomotor efficiency, that there is evidence that the wider the pelvis, the more energy it takes to walk a kilometer. And so that is the selection pressure that is producing that compromise between pelvic widths. A wide pelvis is good for birth, but you need a narrow pelvis for locomotion.
Thank you very much.
More questions?
There's one at the back there.
Okay. As my colleague said and Maria said, first of all, I want to congratulate you for this wonderful presentation. Thank you so much for being here talking about these very interesting topics. But since they are very interesting, they are also very intriguing for us. So we have a lot of questions, or at least in my case. So I'm just going to mention one, but maybe I will ask you later more. So in terms of sperm biology, you were describing the correlation between like sperm volume, sperm number, even motility, and there was a very clear correlation of fertilization of the egg and the sperm number, for example. But I wanted to ask you if has anyone tested if there's like intra-sperm competition for getting into the egg? So, I mean, the 2 million sperms, you said that there are like biological filters in the vagina, but do my 2.5 million, not my specifically, but overall, do the 0.5 million sperms compete with each other to get to the egg? Or it's just the fastest is the one that gets first and fertilize the egg, sorry. And that's one of my questions.
Yeah. You know, some people have said that if you look at a person, you might say to yourself, that is the sperm that won the race. But some people have said that if you look at somebody else, you might say to yourself, that is the sperm that won the race. And there's a fixed idea that there is a competition between sperm. I don't think that is possible because the hereditary material is inactive. So where is the mechanism for sperm to be selected? It's not going to affect their swimming speed. I mean, we can't say that the fastest swimming sperm has some kind of genetic advantage. You know, it might do, but it's a very primitive criterion to suggest that the fastest swimming sperm is somehow the best. And as I tried to point out, I mean, the female tract is designed to slow down and spread out the sperm so that you don't get too many around the egg. I didn't have time to go into the fact that in the cervix, in the neck of the womb, there are crypts. And something like 10% of the sperm don't go straight up to the womb. They swim into these crypts. And they are released over a period of at least 10 days. I think maybe 2 weeks. And this is something that has really not been considered in the medical community, of sperm storage in the tract. But what this means is that some sperms are going to these side tracts and will be released maybe 5 days later. And yet, it depends on the time of ovulation. The first sperm swimming up, if they don't meet an egg, they end up in the abdominal cavity, functionless. And so it really, the timing depends on when the egg ovulates from the ovary and the sperm coming up. So it can be one of these delayed sperm that is released from the neck of the womb that actually fertilizes the egg. That is not a fast-swimming sperm. And I really can't see how there could be any advantage of any particular sperm. I think it's random, like a lottery. There's some evidence that the more crossing over you have during cell division, during the production of eggs, there's exchange of material between chromosomes. And the more that occurs, the larger the sperm count for that species. And so I see this as a lottery, and if you have more variety being produced by this rearrangement of chromosomes, you print more lottery tickets.
May I have time for another question? Or if anyone
He's
else
asking
wants.
whether he can make another question.
Yes.
Okay, thank you. But that goes to the third part of your topic. You were talking about the menopause in humans, but I was thinking about in humans we are kind of, because I was surprised but that the human bar got to 120 years, something like that. And in the non-reproductive period of humans, like modern medicine is interacting a lot, because probably we get that long in the age because of modern medicine. And I wanted to ask you if in like older, like Pleistocene or whatever, or even like modern populations not medicalized, if these non-reproductive periods gets smaller, and it could be more similar to other primate species that you showed in the upper and medium bars. I don't know if this makes sense or...
Yeah, so it's mostly referring that when we see menopause and this very large span for the non-reproductive period, may be an effect, the fact that medical advance is making us living like much longer until 120, but maybe we don't have this human intervention in the lifespan, maybe it would be similar to other animals.
Right, yeah. Yeah, I didn't have time to explain that properly. My interpretation of menopause is that any mammal that reaches about 50 years of life has reached the maximum capacity for limiting mutations, and that it doesn't make sense to reproduce beyond that, not because of a grandmother effect or a grandfather effect, but because the degree of genetic change has simply become too dangerous, and that any baby born is more likely to suffer some kind of inhibition. And the question about medical intervention, I mean, there are cases now where using artificial approaches, a woman of 60 or even 65 can become pregnant and give birth. And it depends whether the egg is a 50-year-old egg or whether it's a 65-year-old egg. And provided that, I don't think that reproductive specialists are aware of this problem, but the thing is, if you take eggs from a 20-year-old woman and freeze them, if you unfreeze them when she's 50, and you then implant the fertilized egg in the womb, genetically speaking, that is an egg of a 20-year-old woman. And so that kind of possibility has to be borne in mind. But if medical science tries to extend the reproductive life of a woman, then they're going to run into problems of accumulated mutation. Okay?
So if I may make a little question before we get the rest. You were mentioning, and I think it's very interesting, that this grandmothering hypothesis would be also covering grandparents, because if not, they would be dying at the age of 50. But if I understood correctly, and I think I read this in one of your posts, which I make use of the opportunity to recommend Bob's post in Psychology Today website. Is a very interesting web blog he has. There are some animals with menopause, not primates, where males do die much earlier than females, isn't it?
Absolutely. Yeah.
So it would be a different case in that, no?
I didn't have time to go into that either, but as I said, there are several whale species, dolphin and whale families, that do have menopause. And this is true of the killer whale, the orca. Okay. And so a lot of field work has been done on killer whales now to try to explain this, and people have tried to support the grandmother hypothesis. And so female killer whales can live to 100. Most of them live at least to 80. So they're living for at least 30 years. They enter menopause at 50 years of age, like women. And so what they've discovered is that in times of food scarcity, there are populations that primarily feed on salmon. And what they found is the older females, the grandmothers in the group, take a leadership role and seem to know where to go for the best salmon preying when food is limited. But the thing that Maria mentioned, which nobody talks about, is that male killer whales die at 50. Okay, and so menopause in whales is very different from us. In killer whales, you see what I was suggesting, males die at the age of 50. They're no longer of any value. And why doesn't this happen in human species? You cannot apply the grandmother hypothesis simply to the killer whale and to us. There are real differences.
Thank you so much. So I think it's time for you to rest and to enjoy the party we have prepared for our birthday and for your moment. And before giving you the final clap, we would like to use the opportunity to give you a gift. This is a special for you to remember this, your visit to us. And
yeah, this is the one, the only hand axe.
Yes, that's the hand axe.
This is from CENIEH Los Rezos.
Yes.
Yeah, this is the only stone tool from CENIEH Los Rezos. I learned today from Juan Luis when he took me around the museum. This is a cast of the only stone tool that is found in the site of CENIEH Los Rezos. And so I've looked at the original of this. It's red. It's a reddish original stone, and the suggestion has been that it might have played some ritual purpose because there are no other stone tools in that particular site. But I never dreamed that I would be able to take a replica of that back home with me. So thank you very much.
Thank you to you. Thank you so much for today. Thank you. So now you are all invited to join our birthday party. So please help yourself.
Yeah, this is
Yeah, this is
such a nice
now
present.